A very good evening to all of you. Welcome to today's session on the gut microbiome, decoding health and disease with advanced sequencing. The gut microbiome has emerged as one of the most fascinating frontiers in biology, influencing everything from immunity and metabolism to neurological health. With advancements in sequencing and bioinformatics, we are now beginning to decode how this complex microbial ecosystem shapes our well being and contributes to disease. To guide us through this topic today, we are joined by Mr. Shantanu Bafna, bioinformatics engineer at Strand Life Sciences. Shantanu brings over a decade of experience in developing and refining genomic analysis pipelines, from oncology variant calling and CNV detection to machine learning models that predict long COVID and cancer outcomes. In his previous roles, Shantanu has also spent several years leading the development of non invasive prenatal testing pipelines, including VeriSeq NIPT and TrueSight NIPT, and has contributed to Dragon based tools for variant calling and polygenic risk scoring. His earlier research at Henry Ford Health System and UC Davis focused on statistical genomics, disease risk modeling, and recombination inference. With this expertise spanning human genomics, algorithm design, and predictive modeling, Shantanu is uniquely positioned to help us understand how advanced sequencing and computational insights are unlocking the secrets of the gut microbiome. Welcome, Mr. Bafna. We look forward to your insights for today's session.
Thank you. Um, yeah, so hi everyone. Today I'm going to talk about the gut microbiome and share some insights from large-scale metagenomics and integrated sequencing approaches. So today, what we are going to do is move past that simple story of just good versus bad bacteria. Uh, Looking at the metagenomics techniques and the new sequencing techniques that have come up, we can definitely say that it's not as straightforward to just mark them as good or bad bacteria. So today, what we are going to do is we are aiming for a more of a three-dimensional view where uh, initially we are going to look at a massive study where they have looked at bacterial signatures in diseases. Secondly, we are going to look at the influence of diet and the physical gut environment. And thirdly, we are going to look at the workflows that are involved in metagenomics analysis. So what we are going to do is we are just pulling some insights from really cutting edge analysis that are kind of redefining what healthy even means in the gut. So we are going to start with why are microbiome signatures important. If we look at the first figure on the left, it displays like a composition of what the gut ecosystem looks like. So basically, the gut ecosystem is formed by like a super species with a large genome composed of different microbial lineages whose genome contains functionally similar set of genes that would or could give rise to coordinated single metabolic outcome. So here, metabolites are represented by circles. The diversity and abundance level of microbe genes, proteins, and metabolites will influence the energy balance, the gut motility, inflammatory tone, mucosal integrity, appetite, and signaling. Also, the key gut player here is pathogens, which may negatively influence the gut barrier, which might even promote inflammation. If we look at the figure on the right, uh, here we are looking at two different sections. One is homeostasis, one is dysbiosis. So In homeostasis, the gut microbiome is supposed to be at the normal levels, but when we talk about dysbiosis, the gut microbiome is a key factor when we are regulating the host health. So, when generally stable factors like stress, any kind of pollutants, and disease, they sometimes cause changes in the gut microbiome, and this state is called as a state of dysbiosis. Uh, and this state often promotes growth of more virulent components like microorganisms and potentially harming the host. And a lot of research has linked gut dysbiosis to many common diseases like cardiovascular diseases, autoimmune disorders, and some cancers. So, in this slide, um, we are going to look at a study where they have kind of analyzed about 6,300 or so fecal metagenomes from 36 studies. So these are basically like snapshots of the entire gut community or the microbiome. Um, and this was specific to Chinese population. Um, and here what they found out was that uh, uh, there was observed a lot of microbial diversity in various diseases. When they performed the meta-analysis, they identified about um, 277 disease associated species where pathogens were increased and beneficial microbiome microbes were decreased. So basically, what they did here was they built a statistical model based on the random forest classifier um, to mark these 277 species, whether and classify them as healthy or disease sample. So what they saw was that this model kind of demonstrated a solid and a reliable performance where they achieved a area under curve score of about point 0.7 to point 0.8. They classify for classifying like high risk diseases, the performance was even better. Closing close to the point 0.8 range, which then successfully led to the formation of what they call as a GHI index, Gut Microbiome Health Index. So this index is basically a quantitative score for uh, implicating whether the gut is healthy or not. And what they saw was that in this study, people with disease observed a higher score close to 650 or 655 compared to the control group, where the GHI score that they saw was about 491. Confirming that if the GHI score is higher, that means there is some dysbiosis being observed or a disease state being identified. In the plot on the right, they have kind of analyzed the gut microbiome data, and it shows that like in 27 out of 40 case control studies, the presence of disease significantly changed the overall makeup of the gut microbiome data. So if you look at the figure on the right, we can see like an atrial fibrillation, ASCVD, hypertension, Crohn's disease, PCOS, we see a significant R square value being observed. So now, moving forward, we are going to talk about how they observe distinct or identify distinct microbial signatures associated with the disease. So what they saw was that the disease leaves a distinct signature on the microbiome, and uh, a lot of common disease are associated with profound microbiome change. So, looking at the results in both these figures, for a lot of condition, they observed that disease means less diversity in the gut, and that pattern kind of showed up when they were looking at Crohn's disease, COVID-19, and hypertension. But surprisingly, what they also saw was that in diseases like Parkinson's and atrial fibrillation, which is highlighted in red on the right here, that microbiome changes are not necessarily associated with reduced diversity and richness, but it can also be the other way, where the diversity is increased in some cases. But what this basically kind of forces us to think is that it's not always losing the good bacteria. Sometimes it's about replacement as well. So in this figure, they have categorized disease by their microbial signatures. Looking at this figure, we are looking at disease enriched species. In disease enriched species, the gross relative abundance score. Uh, and this box plot shows the um, gross relative abundance of the case control groups um, by these are the interquartile ranges with median indicated by the small line. So when we look at the case control comparisons, what we saw that the significant differences that were observed between patients and controls. So in some cases, the depletion of healthy bacteria and expansion of unhealthy bacteria was observed, um, where there was a significant depletion of control enriched species being observed, and expansion of disease enriched species in cases of Crohn's disease and breast cancer. In some cases, they saw that isolated depletion of healthy bacteria was observed. In the third case, they saw that only the enrichment of unhealthy bacteria was observed in cases like atrial fibrillation and IBS. And um, uh, 12, there were 12 disease groups or 12 case control comparisons in these disease groups where no significant changes were observed in either healthy or unhealthy bacteria. But now we are going to talk about the influence of diet and environment on the gut microbiome. So here we are looking at this figure. which identifies the key drivers of microbiome variations and how the gut physiology kind of shapes the microbiome. So, if we look at these six groups, the first one is beyond diet. Like, so even when people are eating similar types of food, their gut microbiomes can differ significantly, which in turn indicates that there is a factor beyond diet that strongly shapes these microbial composition. Then there is gut transit time, which is basically the rate at which the food moves through the digestive tracts, and that also affects microbial activity. So the slower transit time allows more protein breakdown and methane production, which also in turns the gut environment. The third being the gut pH environment, where we are looking at the acidity level of the gut, and that determines whether bacteria can thrive or not. So lower pH is often caused by short chain fatty acid production, which suppresses acid intolerant microbiomes and kind of supports beneficial ferment. Then the fourth is stable individual baseline. So each person maintains a stable gut environment with a consistent pH and transit time, and it is unique for every individual. It can be a little acidic for some. In some cases, their pH baseline could already be very low. So it depends, again, very personalized. Um, then depletion of beneficial microbiomes. So the control enriched bacteria, which is dominant in healthy individuals, are usually found to be depleted in the disease groups. Um, and this groups includes like key short chain fatty acid producers like Roseburia, Plotia, which generates butyrate and supports the internal lining in the gut. And the last being enrichment of pathogens. So, in disease states, it is shown that there is a increased in disease-enriched microbiomes and opportunistic pathogens, which are often linked to inflammation, infection, and even some of the cancer pathways. So moving forward, we are going to go through some of the common workflows that are performed when we are analyzing microbiome data. So this is how the basic workflow looks like. The first step is sampling and DNA extraction, followed by sequencing. Then once sequencing is performed and reads are generated, bioinformatics analysis is being done to look at how the data is being processed and generating insights, and then ultimately visualizing and trying to interpret the data from bioinformatics analysis. So firstly, we are going to talk about sample collection and DNA extraction. So in this step, the goal is to obtain high quality DNA from the microbial community within a sample. So the sample is gathered from various sources, whether it is fecal matter, saliva samples, swabs. Um, there is virus-like particle enrichment, where we separate viral particles from samples. And this involves a lot of steps like filtration, centrifugation, and chemical treatments, and to purify the viral DNA. Then there is DNA extraction, where DNA is extracted from the raw samples or the enriched virus-like particles, and a lot of kits are available to perform the DNA extraction. So the next step in this workflow is the DNA sequencing analysis. So on the right, I have two figures. In the first case, we are going to look at uh, different sequencing, amplicon versus shotgun sequencing. Um, so when we compare amplicon and shotgun sequencing, we um, look at like if it is target specific, like if it's only targeting a specific marker or genes, then we prefer amplicon sequencing, like 16S rRNA gene for bacteria, or the ITS region for fungi. And when we are looking at shotgun sequencing, this method basically sequences all the DNA fragments in the samples and provides data for both taxonomic diversity as well as functional analysis. The second figure on the right, here we are looking at different sequencing platform, and this figure kind of gives a brief overview of long and short read methods, and looking at advantages of each technology. So the first figure here is the PacBio sequencing technology. Here, the DNA fragment in PacBio is ligated to hairpin adapters to create basically a topologically circular molecule, which we can see here. And this is called as smart bell. It is loaded on a smart cell for sequencing, and the same DNA molecule is sequenced multiple times, and every iteration kind of produces a read, which may or may not contains some error in some of the bases. And these circular consensus algorithms analyze and merge reads from multiple iterations to remove these errors. That's why they do it in an iterative fashion to remove the bias and the error, and produce a very highly accurate read data. The second figure here is the Oxford nanopore sequencing. Here, the DNA is tagged with sequencing adapters, which, if you look at the adapters, which is marked by the brown dashes here, um, these are preloaded with like a motor protein on both the ends. A single DNA molecule is passed through a protein pore, and as the molecule passes the pore, it changes the electrical current in a distinct manner, and it is determined by each nucleotide. And this disruption is then measured to establish the genetic sequence. The last sequencing platform here is the Illumina platform, where the DNA is fragmented into smaller pieces and then linked to adapters, which is followed by library preparations, and individual DNA molecules are sequenced to generate short reads. So now we are going to look at some of the limitations of long and short read uh, platforms and how a hybrid approach is being used to overcome the limitations of these two platforms. So in case of short read sequencing, like we saw here, the Illumina platform, it produces reads typically up to around 300 base pairs. Uh, this method is established to kind of generate a vast amount of data. However, what we also see is that assembly using only short reads has some challenges. Firstly, because the reads are short, and what it means is that it often results in fragmented genomes and can lead to a under estimation of the diversity of the virome. And uh, viruses that were assembled exclusively using short read methods um, kind of represents a very smaller portion of the compiled catalog compared to long read sequencing. So in cases of long read, like PacBio and Oxford nanopore techniques, or the platforms that we saw, it basically generates a longer DNA read, and the length is about 1,000 base pairs. Some of the key features of long read sequencing are the length advantage, the assembly improvement, and the viral discovery. So by viral discovery, what I mean is that long read sequencing, it is proficient at recovering like low abundance viruses and sequences from viruses with large genomes. And although this is much better than short read sequencing, but here also there is a cost factor that comes with it, and sometimes it also results in unstable sequencing quality, which leads to the combination of long and short read sequencing techniques, which is the hybrid method, where they combine the PacBio long read and Illumina short read platforms to assemble longer and more complete gut phage genomes. And they have also observed that it this approach recovers more complete viral genomes, close to 35%, that on compared to short read only databases, which is about 4%. And about 60% of the new viral OTUs were assembled using long read or hybrid method, but some overlap with short read assemblies. And this method uncovers a huge hidden diversity and includes thousands of novel viruses missing from the pre-existing database. Now we are going into the deep dive about the bioinformatics analysis and the steps that are performed in metagenomics or microbiome analysis. We are going to start with data processing. So here, for short reads, there is a tool called as FastP or Trimomatic, which is used to remove the adapters and low quality reads. In case of long reads, they are corrected using uh, the CCS tool. And in case of amplicon sequencing, there is a tool called as vSearch, DADA2, or Tippler. The next step in this analysis is to remove the contaminants um, and to basically ensure that we have a cleaner data. And this is a necessary preliminary step in the analysis. Uh, so the basic purpose here is to eliminate the contaminating sequences uh, from the data set, primarily human host contaminants, and secondly, the bacterial contaminants. So the human host contaminants are often identified by aligning the reads against the human reference genome using bowtie 2 tool or software. And in case of bacterial contaminants, this is achieved by mapping clean reads to the databases of bacterial genomes and removing the aligning reads. The third step in the bioinformatics analysis here is the genome assembly, where we basically reconstruct longer continuous DNA sequences from whether it is short, fragmented reads or long reads coming from the sequencing technology that is being used. And this is one of the critical steps that occurs after QC and the contaminant removal. Uh, and some of the softwares that are being used to perform the assembly are IDBA, UD in case of short reads, Canu and Fly for long reads, and Opera MS and Meta Spades for hybrid reads. The fourth step is kind of classification and quantification. Um, or identification and assessment, which verifies contig or genome type and its quality. So in case of identification, this is an initial step of profiling where genetic sequences are classified against specialized databases to determine the identity of what kind of microbiomes are present in the data. And the second being the assessment, where um the identified taxonomy or taxa are measured and interpreted, which includes quantifying their relative abundance and examining what is the diversity being observed in the microbiome data. So this is followed by profiling. The first profiling technique we are going to talk about is the taxonomic profiling, and it is one of the major uh, steps when we're looking at the analysis within metagenomics. And this is used to kind of determine the microbial composition of a community or a structure of a sample. If you look at the first figure on the right, here is a example figure where they have identified and based on three different sequencing, whether it is short read, long reads, or hybrid, what kind of phylum was observed. And it also kind of involves classifying sequences against these databases that we talked about. And in case of functional profiling, um, we annotate protein or genomic sequences using tools like HMM scan and eggnog. The ecological and statistical analysis is the last step in the bioinformatics workflow, which focuses on characterizing microbial communities and their environments. And some of the key metrics that are included in case of ecological analysis are the diversity, composition, and environmental links. By environmental links, what I mean is how the gut physiology shapes the microbial composition and metabolism. Um, in case of statistical analysis, we apply a lot of quantitative tools to validate the findings that we saw and build models. Like in the previous example, they had what we looked at, they had used the random forest classifier. So basically, such classifiers are used to identify significant differences in diversity or abundance between disease or healthy groups. And these are some of the tools that are being used for whether it is clustering and phylogenetic analysis like FAST-3 and muscle for statistical modeling. For MANOVA is used. And statistical modeling, most of the packages are kind of available in R. For classifiers, there are uh, several bioconductor libraries available like Random Forest and PROC. Uh, so in this slide, what we are looking at in the previous section, we saw all the bioinformatics steps that are involved or the complete workflow that is involved in the analysis part of things. And at Strand, we provide a very personalized approach from scratch that provides precision and kind of insights that you need to advance your research when it comes to microbiome data. And we offer several um, solutions, whether uh, it is providing like a comprehensive microbiome data analysis. If you're looking at internal and external data sets, how the data needs to be harmonized to be used for the workflow, and different studies based on the samples being used, the sequencing techniques being performed requires specific kind of development or more tailored development, I would say. Um, we also offer like computing solutions for large scale data sets, some visualization interfaces based on how you want to see and view the results of the analysis, or look at the interpretations of the data and bioinformatics support, overall bioinformatics. Thank you.
Thank you so much, Shantanu. I would request the audience to put in any questions they might have in the Q&A box. We have a little bit of time to take some questions. All right. Looks like we don't have any questions for today. Thank you so much, Shantanu. It was a very insightful session today. And thanks a lot to the audience. I see there's a hand raise here from Shreya Maiti. Shreya, would it be possible for you to drop your query in the Q&A box? Okay, Shreya has a question. In case I have an in vitro fermentation sample with mice fecal sample as inoculum, can I get both taxonomic and functional profiling done?
Yes, yes. So for like it, it is based on the assembly that is being used in case of uh, humans. We use different genome reference. In case of mice also, there are multiple reference data sets available which can be.
Thanks, Shantanu. Shreya, I hope the question is answered. Do we have any other questions? How effective is that data as compared to the fecal sample metagenomics?
Mm, like how effective is the mice sample data or
Hi, Shreya.
Hello.
Shreya, yes.
Oh, okay. I had this question. Suppose I have taken fermentation samples for metagenomics for both taxonomic and functional profiling, as compared to I took the actual feces as my sample. So definitely there would be some changes in the consortium, right, in case of fermentation as compared to taking feces as the sample. So how effective is that data? Because as we know that anaerobes die in contact of air or something. So due to the handling issues, is there a chance that my uh, if I get a profiling done, so both type of samples would be the data for both the samples would vary a lot. So
uh,
is there?
maybe because I think one of the main challenges when we are uh, working with microbiome data is the handling of the samples, how they are being transferred, how they are being kept. But there, I would assume there would be definitely some difference if the samples are damaged. But uh, yeah.
Okay, okay, thank you.
Thank you, Shantanu. Looks like we don't have any other questions for now. And thank you so much for your insights in this session. And a big thanks to the audience for tuning in. If you have any further questions or follow-up questions that you would like to ask, please feel free to reach out to us. We can facilitate and get some replies for you at the earliest. Thank you so much for joining us and have a great evening. Thank you, Shantanu.
Thanks. Thanks.